this video, we will be looking at the replication of DNA. DNA is the genetic material contained within the nucleus of all living cells. Cells replicate to generate growth or for tissue repair. Each new cell needs its own set of genetic material, and this is why DNA needs to replicate. DNA is made up from nucleotides: a phosphate, a 5-carbon sugar, and a nitrogen-containing base. In this demonstration, I will show you how DNA replicates so that each new cell has identical genetic information to its parent cell. This is done through a process called semi-conservative replication. We have evidence of this by taking advantage of the different physical properties of two isotopes called light nitrogen and heavy nitrogen. In the actual experiment, bacteria with DNA incorporating light nitrogen are placed in a medium of nutrients, including bases incorporating heavy nitrogen. After a period of time, replication takes place, and we can see that new DNA in the bacteria cells contains heavy nitrogen, and the following demonstration will explain how this happens. Let's imagine this DNA is our initial sample of bacteria. It is made up of bases incorporating light nitrogen. Both strands of DNA in the helix are green, representing light nitrogen. The bacteria are now in a medium of nutrients incorporating heavy nitrogen. When replication takes place, the two strands unwind and become individual strands of light nitrogen DNA. Various enzymes including DNA helicase and DNA polymerase and the presence of ATP make this happen. We have two individual strands that now need to complete the replication process. During replication, new nucleotides are created and the bases incorporate heavy nitrogen. The new DNA is made up of light nitrogen bases, shown in green, and heavy nitrogen bases, shown in yellow. We can see which part is the original DNA from the bacteria and which part is newly manufactured, but let's take a look at what happens in the next replication. Once again, the DNA unwinds, and we have two pieces of DNA made up of light nitrogen and heavy nitrogen. When the unwinding is complete, we see that we have two strands of the original DNA in green and two strands of the synthesized DNA in yellow. Replication takes place once again, and we are in a medium of nutrients containing heavy nitrogen. Each original green DNA manufactures a heavy nitrogen counterpart, and the manufactured DNA does the same. We now have 4 new pieces of DNA. 2 pieces incorporate light and heavy nitrogen. This shows that one strand of DNA is original in green and the other strand is manufactured in yellow. The other 2 manufactured strands from the previous replication have now formed more new DNA, as we can see. The DNA helix is made entirely from manufactured DNA. We will complete the cycle one more time. After this replication, we can see the original DNA has become more scarce, but it still exists, whilst more of the replicated DNA is manufactured. The presence of intact original DNA strands is why the process is called semi-conservative replication. It also shows how original DNA remains intact even after a number of cycles. The ability to replicate and build new strands of DNA is due to the nature of the base pairs in the nucleotides. This ensures that through successive replications of DNA, the genetic material remains unaltered, except in the case of errors which become genetic mutations, but that's a separate issue. Here's a thought. Your original DNA is made up from the genetic information from each of your parents. The original DNA was contained within a single cell that started you on your journey to becoming a living person. Over the months of gestation and the years you have been alive, virtually all of your DNA in your bodies has been manufactured. But somewhere in your bodies there may still be 2 strands of original DNA that actually originated from your parents.